to form our biologically active unit. So they are subunits, those protein chains. And if all the subunits are identical, that's called an oligomer. If it's two subunits, it's called a dimer. That's what my tubulin was, a dimer. Not as common as trimers and tetramers, but we know hemoglobin is a tetramer. It has 4 subunits that come together. And again, the same interactions that put together our tertiary structure are going to put together our quaternary structure, our subunits. I believe that you can have your disulfide bonds as well. Yes.
Did you say the one was a ligamer?
A ligamer, if it's two of the same subunits coming together, that's a ligamer.
Okay.
Okay, so proteins with quaternary structure, there must be some reason why nature has put subunits together. Why didn't they just make it one big chain? Well, it's because they often can be what we call allosteric, where one subunit will bind to a molecule, and that molecule that it binds to is called a ligand, and cause a change in the structure. We see a conformational change in other subunits that makes it either more receptive or less receptive to a molecule. And we see this kind of behavior in hemoglobin. A little bit about hemoglobin first. We know it's to transport oxygen where? In the blood, right? It's a tetramer, and it has 2 different types of chains, 2 alpha chains and 2 beta chains. This is supposed to be alpha and beta. And each of them, whether it be alpha or beta, has a heme group. So there's 4 heme groups. Each one can bind a molecule of oxygen. And what we see is positive cooperativity. When one oxygen binds to one of the subunits, it becomes easier to bind other oxygens. There's a conformational change. Once one oxygen comes in and binds to one of the subunits, that changes the conformation of the other subunits, and it makes it easier for the next oxygen to bind. That means that the structure of hemoglobin, HB is for hemoglobin, without oxygen is different than the structure with oxygen. There's other molecules that can affect the ability of hemoglobin to bind oxygen. And we're going to talk about the protons, the carbon dioxide, and BPG in a moment. All right. So here's our hemoglobin. I get to have the nice colors. You guys have not such nice colors. So the blue are the alpha, and the green are the beta subunits. And you can see each one has a heme. All right, we're going to spend some time on the next few slides. But the binding of hemoglobin for oxygen is decreased by the presence of hydrogen ions. Protons cause it to bind poorly. Also, carbon dioxide cause it to bind oxygen poorly. And BPG also makes oxygen bind to hemoglobin poorly. BPG is a very interesting molecule. It is there to make sure that we get rid of every last bit of oxygen on our hemoglobin. So you see this molecule in our blood, and what it will do is it will basically kick off any oxygen. Pregnant women have a 30% increase in BPG over the rest of us. You don't need to know what these C1, 2, and 3. Don't worry about those. And interestingly, fetal hemoglobin binds BPG to a lesser extent because it's going to get it. So the mom has a lot of BPG around. The BPG is going to come in and kick out any oxygen that's on the hemoglobin. The oxygen will be floating around, go through to the baby. The BPG will not affect the binding of oxygen to the baby hemoglobin. So that's how oxygen gets from mom to baby. Because if they had both the same affinity for oxygen, there'd be no reason for it to go from the mom to the baby, right? Babies take whatever they want. They will drain your bones of calcium and of every nutrient and every last bit of energy. And you're left with nothing. And they come out nice and healthy. Evolution at its best, right? It leaves the mom ragged and come out with a healthy babies. I had very heavy babies. All right, so let's take a look at this. What happens when we have this like axis in the middle between our alpha and beta, some sort of symmetry there. And when we bind, we have this turning of 15 degrees of this axis from the one alpha beta section to the other alpha beta section. So it's a turning. They slide by each other. And that's because salt bridges and certain hydrogen bonds are ruptured. But next what we're going to do is talk about how pH affects the binding of oxygen to hemoglobin and how it's different than the binding to myoglobin. So there's two concepts that we're going to cover. We have a very busy slide. Next slide. So what we say is myoglobin binds to oxygen normally, while hemoglobin shows what we call cooperative behavior. So what we mean by cooperative behavior is that each oxygen added to the heme makes it easier for the other ones. Myoglobin only has one heme, so there's nothing else going on. It binds to oxygen and it lets the oxygen go. It doesn't really do much. But because hemoglobin has 4 subunits, each with its own heme, it has an effect. Once you bind the first one, it's going to affect the other one. So we see this in the binding curves in their shape. And this is really important, guys. If I start talking about something and you're not getting it, you need to be vocal right now so that I can have you with me. So myoglobin curve is what we say hyperbolic, which I'll get to in a second. And the hemoglobin curve is sigmoidal. So again, I said this slide is very, very busy. So we're going to spend some time on this. Have you guys seen this before? This slide, the oxygen binding slide, or a curve? No? Yeah? So in other class, what class did you see it in?
Bio 2.
Bio 2?
Paramedic class. So.
Okay. So we see this shape as hyperbolic. All right? This myoglobin, that shape I want you to recognize is hyperbolic. And the hemoglobin shape is what we call sigmoidal. Looks like an S. That's easy for me to remember. I remember myoglobin is hyperbolic because it's hyper. It goes up quick. Right? That's how I'm going to remember that. And then the sigmoidal shape comes from it looking like an S. All right, I want you to just take a look at the myoglobin. On the y-axis, we have the saturation with oxygen. What does that mean? Here, this 0.5 means that half of the myoglobin molecules, proteins, are with oxygen. This 1 means all of them are. So that's what the y-axis means here. And the x-axis is just concentration of oxygen. So we're going more and more oxygen on the x-axis. So let's spend some time with just the myoglobin. We can see that it doesn't take very much for our myoglobin, very much oxygen for our myoglobin to get filled with our oxygen. But as we go up and up, what's happening? It's harder and harder, right? I am just focusing on the myoglobin. I don't want you to pay attention to the hemoglobin. Yeah. But here is our myoglobin. So come in and essentially it starts filling up and then it's harder. Okay, there's one thing I want you to keep in mind, that molecules are stupid, okay? All they do is they bounce around randomly, okay? They don't know, hey, I already bounced into a myoglobin that has oxygen, right? So it can't look for something. So now I want to take a trip to Crossgate Mall. And imagine the parking spots are myoglobin. So say we have 100 parking spots, 100 myoglobins. And the cars are the oxygens. And we have 100 oxygens. If you're the first one of the cars, you can find a spot pretty easy. You're parking right next to Old Navy, right? You're right there. That's where I park. And I have to go to the mall, which is not a fun thing for me to do with teenage girls. No. Okay, so the first one in, you're going to find a spot. Hey, the first 50. It's pretty easy. But what if you're like the 80th person? Is it harder to find a spot? If there's only 100 spots and you're the 80th person. But you're smart, right? You're not going to try to park on a spot that's already filled and crash, right? You're smart, right? You're going to look for an empty spot. So, but oxygens are not smart. They're going to go bouncing around until it finds an empty spot. So that's why you see this shape in this curve. This is why we don't just go straight up to 1. You see this curve because these last 20 out of the 100, so we're at number 80 oxygen, it's harder to find an empty myoglobin. So right here, maybe in your notes, up here, harder to find that empty myoglobin. Why doesn't it exactly get to 1? Because that last one is bouncing around a long time before it finds that one empty spot. Remember, it doesn't know. I've already been down this lane, and I see that they're all filled. Okay, I know you guys do that, but oxygen molecules don't. I know that's really weird, but it works. Okay, so you guys got this shape? It goes up quick because there's a lot of empty spots. Then it slows down because we can't find as many empty spots. And they're stupid. All right. Now, hemoglobin has an S shape. Down here, it's different from the myoglobin, right? And up here, it looks more like it. Do you guys agree? That down at the bottom, at low oxygen concentration, it's harder. And then here, it's a lot easier. So I would say for 1 out of every 4 parking spots, you got to pay a toll for the hemoglobin. And then the 3 other people can come by. So the first one has to pay a toll, and then 3 other people can come in and bind easy. So that's, and what I say, and this is really on the molecular level, I call this hard binding, and I call this easy binding. So down at the bottom, it's hard binding. It's hard to bind. And then up at the top, it's easier binding. It's all easy binding with myoglobin. But here, on a molecular level, it takes more than 1 collision in order to get the oxygen to bind to the heme. Here, it might take just 1, right? Because we've changed the conformation. Once we get the first one out of the 4 hemes in there, we're changing the conformation so then it becomes easier. So you can also put in your notes down here, this is the first one out of the 4. And then here, up at the top, we've changed that conformation to make it easier binding. So again, down here, hard binding. Up here, easier binding. It's hard binding because we haven't changed the conformation. The binding of the first one makes it easier for the other ones to bind. So we need more oxygen around, a higher concentration, in order to get the same amount of binding than we do with myoglobin. Yes?
Does the conformation change each time oxygen binds? Or
No, just once. Just once the first one's on. Then the conformation changes, and then the other three can come in. It doesn't change after the second and then after the third. No, just the once. Okay. Everybody good with this one? All right. This one is a totally different concept. I don't know why I decided to put them all in one slide, but just to make you write tiny. This is called the Bohr Effect. And this is why we can breathe. I want you to notice what happens to the binding or the amount of hard binding as we increase pH. This is all hemoglobin. Again, the axes are the same, percent saturation and the concentration of oxygen. What happens as we increase pH? Does it make it harder to bind or easier to bind? 7.6 is higher, so we get more binding. So let's look at 60%. We only need 20 millimeters of mercury to get 60% saturation, where at 6.8, we need almost 50 millimeters of mercury of oxygen in order to get 60% saturation. So we need less oxygen around at this higher pH to get oxygen in there than we do with the lower pH. So does it make it easier to bind with higher pH or harder to bind? Easier. It's easier to bind oxygen to hemoglobin at higher pHs. Another way to say this is that we reach the same percent saturation at a lower concentration of oxygen at higher pHs. Yep. We reach the same percent saturation of oxygen in our hemoglobin with lower concentrations of oxygen at higher pHs. I can say that again. We reach the same percent saturation, say 60, with lower concentrations of oxygen at higher pHs. So we only need a little bit of oxygen around at pH 7.6 to get to 60%, where we need 50% or 50 millimeters of oxygen at 6.8. And that's not that much of a difference of pH. That's a huge change. Yes?
Can you say it one more time?
Sure. We need let's say, to reach the same percent saturation, we need a lower concentration of oxygen at a higher pH. That's what this plot is telling us. So therefore, we can infer that it is easier at higher pHs to bind oxygen to hemoglobin. This is not a huge change, even though it's a log scale, a huge change in the hydrogen ion concentration. But there's a very marked difference in that. This is what we call the Bohr effect. Same to do with the carbon dioxide as well. So if you have carbon dioxide and hydrogen ions around, you are going to drop oxygen. If you don't have hydrogen ions and carbon dioxide around, you're going to pick up oxygen. You don't have to write, to answer this question on the test, you don't have to write down this this. I need to know that you understand that at higher pHs, as in the lungs, it is easier to bind oxygen to hemoglobin. So in the lungs, hemoglobin will bind oxygen and release our proton, as well as carbon dioxide. And we breathe it out. Breathe out our carbon dioxide. Protons are a product of metabolism, as we'll see later. And what happens is it makes working muscles have a very high hydrogen ion concentration. So since there's a high hydrogen ion concentration, those are going to go into the hemoglobin and kick out the oxygen. So then the oxygen's there for our actively metabolizing cells. The way we get rid of the protons in our lungs is to combine with bicarb, supposed to be a negatively charged on there, which makes carbonic acid, which we can break down to water and carbon dioxide and breathe out. Okay, you guys good with this? You've heard the Bohr effect before? Was it ever explained in this manner? No. So I think we go over a lot of the same ideas as you do in other classes. We just explain it in a different way. Hopefully not conflicting. Let me know if it conflicts with anything that you've learned before. Yes?
The bicarbonate is negatively charged?
Yeah, the bicarbonate here, HCO3, should have a negative charge. I think I tried to put it in there, but it meshes in with the arrow. See, I have it over here, but here I messed it up. So that combines to form carbonic acid, and then that can be breathed out. This is the same thing that happens in reverse in our oceans. This is why we're worried about the acidification of our oceans from greenhouse gases, because carbon dioxide will react with water to form carbonic acid, which releases protons and makes the ocean more acidic. Not to get political. The scientific protein folding. Many people's lives are dedicated to protein folding and studying this. There's graduate classes just on protein folding. Books and books and books, just talking about predicting how a protein folds. You can and you can't predict. I told you the story about tubulin and how the people came out with the X-ray, and we all whined saying that's not true because we had to have the solid to have the X-ray, and tubulin is such a big protein, as you can see here, that it needs to be in its aqueous environment in order to be native. So there's limitations to predict it, but there's a couple different things that we do. The bioinformatics is where we integrate computers with biochem. And I remember when I was in grad school a long time ago, this was a new field. And we had a postdoc that was, this was her job to try to figure out, using bioinformatics, what the tertiary structure of our tubulin was. And so what we do is we do a couple things where we are looking at something called sequence homology. So if you have, say, 25 to 35% the same amino acid sequence as another protein, you can hypothesize that maybe your protein folds similarly. And if you can't really find a similar structure, we try folding recognition. But again, hydrophobic interaction is the major factor in protein folding. So putting the nonpolar side chains inside, and the aqueous environment has the polar side chains around, is the driving force. And again, we're increasing disorder by having the hydrophobic interactions, the hydrophobic side chains inside. Because if we had them on the outside, we'd have that cage of water around them. And of course, I have a YouTube.
So what we're looking at here is a result of the simulation of protein folding from folding at home. We're not looking at all of the atoms present because that would obscure what we're watching. So we're not showing it in the water, and we're not actually showing all of the atoms in the protein either. What we are looking at is mainly the protein backbone, which is that set of tubes that sort of go from red to blue. And we're also looking at the aromatic or a certain type of amino acid backbone, tryptophan, phenylalanine, and so
Those are
on.
hydrophobic.
The reason why is that the aromatic residues form what's called a hydrophobic core, and it's interesting to watch the core form. So what's going on is the protein's trying to assemble into its final state or fold. And it's basically getting some things right and some things wrong as it goes. And basically, as it gets something right, it doesn't, but not completely right, it ends up unfolding. Like right now, it unfolds and loses that structure, but then tries to come back into a state that's better. And so, for example, here it's finally reached the folded state, that the aromatic residues in red and green are basically in their final spot. The blue phenylalanine is flopping around, but that's the way it would be in nature. That part's not structured. It's basically reached the folded state and is perfectly happy to stay like this for a very, very long time.
That takes a microsecond. Is that awesome or what? What we just saw is what happens in nature for that protein to fold, and it takes a microsecond for that to happen. So it folds and unfolds and it folds again until it reaches its native state, and it's those hydrophobic interactions that's going to drive it. You're going to get those non-polar, those aromatic side chains on the inside. Yes.
So was that imaging like made inside of a program, or was that just like a representation of something that they know exactly like how it, like
I don't know how they made that video. I think they have some guesses about what happens during the folding of proteins. They might have seen things and other things that take longer. I don't know how they got that, but the guy sounded trustworthy.
Oh yeah.
No, I think it was really neat. So just to drive that point home, the orange ones or the red are the hydrophobic side chains, and the green over here on the right are the hydrophilic ones. So you can see that the green are all on the outside and the red are all on the inside. Now, when proteins don't fold correctly, bad things can happen. One of the things that we look for in biotech is what we call aggregate. When proteins aren't folded correctly, they clump up together, and you cannot inject somebody with an aggregate of a protein, of a biotech drug. So biotech drugs are proteins, and if you inject somebody with a misfolded protein, you will for sure get an immune response. It will drive their immune system crazy. So one of the things that my job was when I was in industry is to make sure that our product did not have any aggregate in it. And again, the prions, it can be toxic to the cell. You have misfolded proteins, that's what prions do. It will cause death. So, but on the other hand, sometimes we don't want proteins to be active until it's right at the right spot. So what we have are called chaperones that kind of stay with the protein until it's ready to fold. And then the chaperones aid in the correct and timely, or spatially, folding of the protein. Heat shock protein was the first chaperone protein to be discovered. I remember talking about this back before you guys were born. And chaperones exist in all our cells, from prokaryotes to humans.